In order to understand brain disorders and neurodevelopment disorders and their, I guess, molecular and circuit underpinnings, researchers they grew some portion of human brain cells in a dish, and they implanted them into mice. Uh, I'm not going to get into any political side of this or anything. I'm just strictly looking at the science and the results and what details went into it and the outcomes. Some of it's pretty interesting. Some of it makes you makes you think, what is happening here? What kind of monsters are they making? That's kind of like a side story, but basically, they wanted to study mental disorders. This was at Stanford, and they wanted to study mental disorders and brain development. So I guess it's harder to study in a human. It's harder to do this type of experiment in a human. So they successfully connected living human nerve cells, or neurons, and supporting brain cells with the brain tissue of rats to form like hybridized working circuits. Now, to me, just in layman's terms, this looks like a rat with part rat brain and part human brain. And there's a million ways you can go with that. I mean, it's a million ways, but they say they want to study brain disorders like epilepsy, Asperger's, autism, things like that. So in the research, they said that they basically grew manipulated human stem cells. They grew brain tissue through stem cells, right? In one of those dishes. And they wanted to observe the effects of the rat's behavior. So they wanted to study healthy brain development and as well as brain disorders. And they also wanted to study brain disorders. So they took the tissue of this grown human brain. They didn't fully grow a human brain, but they grew some portions. They basically grew them from stem cells, and they implanted them into rats. They couldn't do this with fully grown rats, so they had to do it with rat pups, or basically rats that were like a couple of days old, because with fully grown rats, their brains are already developed. So it would be hard to make the connection with the human grown brain in the dish. Whereas when they implant this cluster of brain cells into a baby rat, that rat's brain is still developing. So they, based on the experiment, basically the rat portion of the brain reached out to the human portion, and they grew together, and they made connections, and they were communicating. And which, again, this is really weird, interesting. I'm sure this has been done a long time with other things, but I mean, you basically have a rat with partially human brain. So now it makes me wonder. I know this wasn't published in this study. Maybe in another study. I know they looked at this. They had to, because you got to be curious. Okay, how are they different now from other rats, right? I know that they say, oh, we want to study Asperger's and autism and epilepsy. Yeah, sure. So now, are these rats more human-like, or what do they? I mean, rats are already smart creatures. They're already, smart at avoiding us, smart at sneaking food. I've seen a rat fight a bird, a pigeon. But yeah, I'm wondering now. Okay, so basically on the experiment though, they say they made basically a complicated circuit in a dish using organoids. Now, organoids are like a cluster of stem cells. Right? They're self-organized three-dimensional tissue cultures. They derive from stem cells. That's what organoids are. So these organoids, they reassemble themselves to look like a cerebral cortex, like the human brain's outermost layer, and its most recently evolved part. Now, the researcher says they've been making more complicated brain circuits in a dish using organoids and sophisticated combinations called assembloids. I'm not going to get into that. It gets complicated, and I want to communicate where I can be understood and where I don't sound like I'm using a bunch of giant words that no one knows what I'm saying. I know it's hard. I probably sound like that now, but I'm trying. Bear with me. So they take these rat pups that are 2 or 3 days old. So this is the equivalent of a human baby. And they perform this procedure in each rat, the same procedure in each rat pup. And they implant these portions, these basically independently grown human organoids, basically part of a human brain, into these rat pups. Again, they couldn't do this in adult rats because their brains are fully developed and it would be hard to make a connection. So after about 2 months or so, when these cells are closely resembling the human cerebral cortex, then they transfer this into the rats, the baby rats. And they replace the organoids, or these human cells, are placed in the same location in each rat. So they want to make it identical. I think I mentioned earlier. So they want to make they want to see how each of the baby rats develop. So once they implanted these cells, the cells from the rat brain migrated into the human tissue. And these cells penetrated that human tissue and they assembled into the blood vessels, which supply like the nutrients and the signaling substances to the human cells, and they take away waste and resident immune cells into the rat brain. They also populated the resident immune cells also populated the rat brain. So basically these two portions of the brain merged right together, and this basically formed into one brain, 1/3 of it being human, 2/3 being a little rat. Again, I'm just imagining. Okay, you have basically a rat with 1/3 of a human brain. I know they're studying brain disorders and development, but does this make them? How does this enhance a rat, or does it enhance a rat at all? I'm leaning towards it probably did enhance a rat in some way. I mean, again, they're already pretty gifted, smart, intelligent creatures. So imagine you got a rat who thinks 1/3 like a human. I don't want to see that outside the lab at all. Mind you, it took 6 months for them to take up approximately 1/3 of the rat brain. So yeah, after 6 months, it took up 1/3 of the rat brain. They did notice that the neurons from the organoids in the rat brains, they grew about 6 times larger than the neurons that were grown in the dish. So the ones that were left in the dish they were 6 times smaller, and the ones that were transplanted to the rat brain grew 6 times larger. So one of the structures where the human neurons forged a direct connection was the thalamus. So that's deep into the brain. So this human brain formed a strong connection with the rat thalamus. And this is the area that relays multiple sensory inputs to the cerebral cortex. Again, this connection is a huge part of the human portion and the rat portion, a huge part of that relationship. So this gets pretty interesting. So I wonder how, again, it kind of shows you how the human part and the rat part relate to each other and communicate with each other. So the part where they want to understand the neuropsychiatric issues and disorders that people deal with, as far as autism, epilepsy, schizophrenia, they look at something called Timothy syndrome. Timothy syndrome is a rare genetic condition that's associated with autism and epilepsy. So they took cells, these stem cells or organoids that are generated from a Timothy syndrome patient. Okay, and this patient was these cells were put into one side of the rat's brain. This is where, again, they want to observe how these brain disorders are function or lack of function, and how they are supported, and what basically gets them going. So they put one side of the rat's brain with the normal cells, and the other side with the Timothy syndrome, autism, epilepsy cells. And they wanted to observe the different sides and how they interact. After about 6 months, they did observe that the cells from the Timothy syndrome patient, their neurons were a lot smaller and a lot deficient when it came to connecting with other parts of the brain. And this is the part where they're supposed to act like an antenna for input from nearby neurons. So this kind of shows that those with the autism and epilepsy, the cells kind of are smaller and deficient. This is kind of what it's, I guess, inferring, right? That the cells are much smaller and deficient when it comes to branching out and making connections. And they say they learned about Timothy syndrome when studying the stem cells or organoids. I'm trying to really keep. I really don't want to use too much complex terms, so I'm trying to keep it as simple as I can. It's kind of hard to. I'm trying to keep it really, really simple. I hope you can follow. So, the cells or organoids that develop from a Timothy syndrome patient, they learn about that when they were kept in a dish. But with this transplant, they're able to see the activity, right? How it communicates with other parts of the brain. Whereas just watching it in a cell, it's not the same thing. So taking these organoids from people, like who have schizophrenia or autism, and compare it to those without schizophrenia or autism, on the opposite side of the rat's brain, they see size difference, complexity difference, and functional and connectivity differences. This is important. This is good. This is a good observation, and I think this is kind of gives a better insight on how to deal with these disorders. Maybe again, they can find a way to improve connections in these patients' brains. I mean, without brain surgery, maybe there are some ways they can help grow these connections or make the connections better. Or who knows? I don't want to. Because I'm gonna go into computer chips into this video as well, but I don't know if they can attach something to improve electric stimulation. I don't know, but I thought this was very good observation and something that would help with figuring out how to cure or treat these disorders. And so they ran several tests. I mean, I could go on and on with this as far as experiment, but I want to also talk about the brain cells that grew in the dish, and these independent cluster of cells learn how to play video games. Yes, this is also this is good and bad. You could go so many ways with this, and I don't want to get too conspiratorial, if that'll even a word. I don't know. They also ran another experiment actually where they did like a reward type of experiment where they flash a blue light, and every time they flash a blue light, they train their rats to go after water, right? So they use this to kind of as a again, I know that they're studying disorders, but what else are they studying here? So the rats learn to associate blue light with stimulation. But I think you can train rats without the human brain tissue. I know that. I've seen that. We've all seen that somewhere on YouTube, of course. But I mean, with food especially. But I'm wondering. They were using the human side. They were focusing specifically on the human side to train the rats, right? Not just the rat portion of the brain. So how they want to see how the human side engages the rats. But anyway, I want to get on this other topic in regards to them growing cells in a dish again, and they basically attach these cells to a chip, to a computer chip, and they attach the cells to a computer chip, and the cells learn how to play the video game Pong. The objective of this is what again? I know I read it, but I'm just thinking out loud. I mean, this is nice and all, but we already have AI. Are they trying to create living computers here? What? I mean, you take networks of brain cells, grow them in a petri dish, and you attach them to a computer chip. And they call this synthetic biological intelligence. Okay. Again, it reminds me of I did a video on xenobots before, I think. If you don't know what xenobots is, it's spelled X-E-N as in no, X-E-N-O, like X like Xavier, xenobots. And they were like cells that were kind of like, again, synthetic in a way, and could be instructed to do certain things. This can go either way again. This can go either way. So now they have a cluster of cells they attach to a computer chip. They show those cluster of cells how to play Pong. Okay, what else do they want to do with this? I mean, we've got algorithms. We've got AI already without the brain cells. Why do you want to attach brain cells to computer chips and create some type of synthetic biological intelligence? You want to create living computers. Why? I mean, Microsoft already has issues controlling its own AI. I don't know. So they basically they dissected some neurons from human stem cells again. They reprogrammed them, and put them into microelectrode array chips. So these chips can record the electrical activity of the cells. And on the chip, the cells mature and they connect with each other on the chip, and they form their own neural networks. And they also have electrical activity, and the chips also stimulate them. And they call it the dish brain system. Yeah, this is appropriately named a dish brain system of synthetic biological intelligence. And again, anyway, the chip showed these cells basically how to play a video game. So they improved every time. It gave them feedback, and these cells basically acted on their own, and they were able to block the ball and play Pong. They learned and improved their performance. Just a portion of a brain learn how to play Pong. So where they want to go with this again? I don't know. I don't know if this is necessary. I mean, I don't know why do you want to chip brain cells? Why do you want to teach brain cells to function? What are you trying to make here? So does that mean something is living? Is it synthetic living? Are you going to create? Anyway, I'm getting into sci-fi. Well, sci-fi is becoming real to us. I mean, it's not just Skynet. Remember Skynet with Terminator? Skynet had no biological cells on it. This was all. It was all algorithms and electronic. So this is like basically human stem cells attached to a computer chip, and it's playing a video game on its own. We already play the computer in our regular video games. We play the computer without brain cells. I don't know if I like this. Anyway, I could go on and on. Let me know what you think. Feel free to leave a comment. Please like and subscribe. And thank you so much for your time.